you stop me if there's something you want to talk about. So blood flow, so here are my answers to this one. Anything to talk about? Pretty straightforward. Which of the following are freely filtered? These three. Labeling. It's just the labels that were in the PowerPoint review. Yes. So, you know, so the pelvis is here. This is a major calyx. This is a minor calyx. Minor, major pelvis. You know, I won't. If you point to that and it's clear, you know that pelvis is on the inside, and major is the next, and minor is the next, going from inside to outside. I'll be good. You'll be good.
How can you tell in a black and white picture which one is the artery and which one's the vein?
You can't. So either I will tell you it's blue or red,
Okay.
or I will not care whether you label it artery or vein. One or the other. But remember, this is systemic circulation. So now, arteries are red and veins are blue, whereas in the pulmonary circulation, which is the trick on the in the exam, arteries are blue and veins are red. Right? That's the difference between the pulmonary circulation and the systemic circulation. Some of you got tripped up by that. Ready?
Is labeling the medulla here as well? Yeah.
So this is trying to illustrate that this layer is the medulla, and this layer is the cortex.
Okay.
This structure is a pyramid, but this layer is the medulla. Other questions?
Would you put afferent artery
No, that's not what they're called. It's the renal artery and the renal vein. We don't get afferent or efferent until we get to this arteriole, the microscopic arteriole that feed into or feed out of the glomerulus. It's true that the renal artery brings blood to the kidney, so you would say it's an afferent artery, but we don't say that. More labeling, lots of detail here. That remember writing it out is useful for putting it in your brain. It's the same picture we used when we were talking through it. So my recommendation for putting this in your brain is to print out blanks and label them until you get it all right. I think I was clear about what nuances are not part of my concern. Thick and thin, convoluted, non-convoluted. It's true that this is called the corticomedullary junction, but I haven't used that. It's not on the key concept list. Abby?
What is the macula densa?
The macula densa is a specialized set of cells of the distal tubule that make the contact with the afferent arteriole, which you don't have to know now. I mean, you can, it's got a name, but the function of which we will talk about next week. Okay, and then more labeling. Again, I recommend printing out a blank and writing in your words. There are some labels that I don't care about. I don't think it's important to learn parietal and visceral, but you know, visceral is on the inside and parietal is on the outside, so you could use that. The parietal layer of the Bowman's capsule, I just call Bowman's capsule. The visceral layer of Bowman's capsule, I call podocytes. Everything else, oh, so we haven't talked about the juxtaglomerular cells. We haven't talked about the macula densa. Those are fair game for, I mean, maybe they would be extra credit labels on an anatomy quiz because I haven't gotten into their physiology yet. So these are going to be important next week, but not yet. And you can see the specialized macula densa cells of the distal tube. You get to say words like juxtaglomerular. Okay, so these are the labels that you should be able to put into your brain. Questions about any of those expectations? Because now what I'm going to do is transition to the next PowerPoint. Obviously, I don't have to do a review because we just did it. I don't have to review the microscopic anatomy. So this is a slide from when there was a gap in time between one class and another. So what we're going to talk about now is we're going to talk mostly about this thing called clearance. And this is not, I'm not sure it's done very well in your book. So this class is your best reference for understanding clearance. That's what we're going to be focusing on. So remember that the glomerulus filters water and small molecules from plasma into a filtrate. The fluid that is going to become urine, it starts out as a filtrate, and then we just call it filtrate, process filtrate or urinary fluid. It's not urine, it's not plasma, it's something in between. But it starts out as being filtrate. Then the tubules, the proximal tubule, the loop of Henle, the distal tubule, the tubules are going to recover from the filtrate, working its way through the tube, the stuff we want. What do we want? We want water, we want salts, we want glucose. Those are the things we want to get back from the filtrate. And that process of getting back, of recovering, is called reabsorption. So I'm going to use that word, reabsorb. We reabsorb salt, water, glucose, and other necessary things, the things we want. And then what we don't reabsorb gets excreted. So we have filtration, reabsorption, and excretion. A reminder of the anatomy, which I don't have to do. A reminder of the anatomy, which I don't have to do. A reminder of the forces, which I don't have to do. But here is new. So we're now talking about filtration still. We can regulate filtration. So now we're talking a little bit about regulation. So let's get this cartoon straight. This is an afferent arteriole, glomerular capillary bed, efferent arteriole. This is the process of filtration. Filtering fluid from a glomerular capillary into Bowman space, and then it becomes the proximal tubule. That's the nature of this schematic. Okay, let's take this figure apart. So the afferent arteriole has smooth muscle surrounding it, just like most arterioles. If we constrict the smooth muscle of the afferent arteriole, we limit flow, we reduce the hydrostatic pressure in the glomerular capillaries because we're constricting the inflow. So if we constrict the inflow, we lower the hydrostatic pressure in the glomerular capillaries, and that reduces GFR. So, GFR, glomerular filtration rate. I'm going to start just using GFR. Glomerular filtration rate, the rate in mils per minute of filtration of fluid at the glomerulus. So, constricting the afferent arteriole or dilating the efferent arteriole. By dilating the outflow, it lowers the pressure in the glomerulus because we're letting it all out the other end and we're not constricting it here. So constricting the inflow or dilating the outflow lowers the pressure in the glomerular capillaries and lowers the rate at which fluid is filtered. And the converse is also true. If the smooth muscle of the efferent arteriole is constricted, or the smooth muscle of the afferent arteriole is relaxed and dilated, this will, constricting the outflow, increases the pressure in the glomerular capillaries and increases glomerular filtration rate. Dilating the inflow, more pressure getting into the glomerular capillaries, more pressure, more force, more higher rate of filtration. Questions about how our smooth muscle on the afferent and efferent arteriole and how constriction or dilation of that could lead to changes, decreases or increases in the rate of glomerular filtration. So
I understand like why if you dilate the afferent arteriole, it would be more pressure. But like, isn't there more volume? So
there will be more flow,
okay.
but the point is it's more pressure.
Okay.
Okay. There will be more flow, but there will also be more pressure. And so you could say, so dilating the afferent arteriole and constricting the efferent arteriole, the flow now is going to be balanced out because we both have dilation and constriction, but now we're going to have a maximal pressure, right? These two could happen at the same time. Why would you want, let's just take a break for a moment, why would you want to increase your glomerular filtration rate? Under what physiological circumstances that you can imagine? CJ?
Maybe you're eating something that's high in whatever your body does, what your body wants to do, like excess salt.
Okay, yeah. So, or what if you just drank a liter of water? I mean, you could drink a liter of water, and now you're getting hydrated, and your body says, "Oh, I'm hydrated. I need to get rid of this." The only way it can get rid of it, the first step in getting rid of water or salt, is to filter it. And similarly, if you are dehydrated, the first step in conserving water is to not filter it. So that's why we would have control over filtration.
Does
So
this work with alcohol? Or
alcohol.
Alcohol has two like processes.
Alcohol is complicated,
Yeah.
But alcohol is a diuretic, typically, it makes you go to the bathroom. And so alcohol, I don't know, I'm not sure I understand the mechanism for alcohol. I think alcohol relaxes the afferent arteriole, but I'm not sure. Yeah,
Are there like different nerve innervations that would make,
there is going to be nervous innervation from the hypothalamus, because the hypothalamus is where we detect the level of hydration. So yeah, we are, yes, there will be some nervous control of the smooth muscle. There's also going to be some local control of the smooth muscle,
okay, so
and
it's
that's kind of
like-
too complicated for us.
it's like the GQ and the GS stories we talked about,
I suppose I could ask you that, yeah,
but like those are the receptor types,
yeah.
like the most.
Yeah, that's true. So we are going to talk about the sympathetic nervous system controlling glomerular filtration.
Okay.
Yes.
Would that be related to like, is it vasopressin?
We're going to talk about vasopressin later. Next week? Yeah, I guess it has to be next week. Yeah, we'll talk about vasopressin next week.
What happens if the afferent and efferent are both constricted or both dilated?
Yeah, okay, so if we constrict the efferent and we constrict the afferent, then we're going to reduce blood flow into the glomerulus. And it's going to balance, in terms of pressure, it will be balanced out, but blood flow will be reduced. And that is going to tend to reduce glomerular filtration. If we dilate the afferent and dilate the efferent, we're going to get a lot of blood flow, and that's going to balance out the pressure, but lots of flow also leads to filtration. Those are tricky questions that I won't throw at you. Okay, so now we have to talk about this thing called clearance, this physiological process called clearance. And some of you may have heard of this, maybe at a doctor's office where you've learned that drugs are cleared from your body, right? You take aspirin or you take, ibuprofen, and I think ibuprofen, you're supposed to not take it more, you take 2 every 8 hours, because the drug gets into your body, and then over 8 hours it gets metabolized and excreted. The process of metabolism and excretion is called clearance. And we have 2 primary ways for clearing things from our body. Renal clearance, which we're going to be talking about, and hepatic clearance, clearance of the liver. The liver metabolizes a lot of things. We are not going to talk very much about hepatic clearance when we get to the liver. The concepts are the same, so I'm going to teach the concept of clearance in the renal section. So, let's unpack this cartoon. Afferent arteriole, efferent arteriole, glomerular capillaries, Bowman space, right? Proximal tubule, peritubular capillaries surrounding the tubule. So proximal, loop, distal is all being just simplified into this tube. So this is the tubule, and this is the capillaries surrounding it. Okay, this is a simplification. Let's see if I can go back. It's a simplification of this. Proximal tubule, loop, distal tubule, capillary surrounding. Tubule, 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 capillaries. Not very far away, really wrapped around. So we have filtration, we have reabsorption, which I've talked about, reclaiming. There is also a minor component where the tubules, the capillaries kind of pump stuff from the capillaries into the tubule, and that's a process called secretion. Secretion, and then we have excretion. What doesn't get, so what is filtered, reabsorption is minus, so We take the amount filtered, subtract the amount reabsorbed, add the amount secreted, becomes the amount excreted. Do not get secreted and excreted confused. They're different. Yes?
What is secreted? Is that like similar to
We're-
what's been filtered? Or is
So we have two ways to get stuff from the blood into the urine. We can filter it, or we can secrete it. Secretion is going to be the smallest component of this story. Filtration and reabsorption are the biggest components of this story. So if you want to limit, if you want to cut yourself to what you need to know filtration, you need to know reabsorption, you need to know excretion, and then secretion is sort of around the edges. So now we're going to do a little bit of arithmetic or mathematics. Well, it's arithmetic. So we have a definition of something called the excreted load. The excreted load is the volume of urine, the volume of what you excrete, urine, in mils per minute. So this should be mils per minute. When we talk about volumes now, we're really talking about flow rates. So even though I use the word V, it really should be V dot. Volume per minute. Milliliters, typically it's milliliters per minute of urine. Okay? It's typically around 1.5 to 0.5, somewhere in there. Times the concentration of something in the urine. So this makes sense. Mils per minute times milligrams per mil becomes milligrams per minute. Or in some cases we do millimolar, or in some cases we do milliequivalents. Some unit of mass or charge per minute. Volume of urine, mils per minute, times the concentration of something in the urine gives us the amount that's excreted in milligrams or something else per minute, and that's the excreted load. So these are the terms that go in there. Okay, so I will ask you to know these equations for the quiz. This will be on the quiz. Or it could be on the quiz. So let's do a clicker together right now. Let's not take a long break to do this. A person produces 1 mil per minute of urine. The concentration of glucose in the urine is 0. That's normal. The concentration of sodium in the urine is 100 milliequivalents per liter, 100 millimolar. Okay. What is the excreted load of glucose? Show me your answers. The excreted load of glucose is 0. 0 mgs per mil times 1 mil per minute is 0 mgs per minute. What is the excreted load of sodium? Now I have to admit that you have to do a little bit of arithmetic. Mils per minute, milliequivalents per liter. Don't let the units trip you up. This one's pretty straightforward, but there's going to be another curveball coming at you later on. What is the excreted load of sodium when we produce 1 mil per minute of urine that was 100 milliequivalents per liter in it? Some of you can do this in your head. Some of you probably need a calculator. B. 100 milliequivalents per liter is 0.1 milliequivalents per mil. 0.1 times 1 is 0.1. Okay, so this is an equation that, on the quiz, I will not ask you to do arithmetic. You do not need a calculator, you don't need your phone, you can keep all your electronics away as you should, but you might be asked to put the equation of something on the paper and maybe the units. I'm not going to ask you typical units, although you now know that you typically produce 1 mil per minute of urine. Okay, so that's excreted load. Now we have to talk about something called clearance. Clearance is defined as the excreted clearance of X. So we always talk about the clearance of something. Clearance of glucose, clearance of sodium, clearance of water, clearance of acetaminophen. You always talk about clearance of something. So the clearance of something is the excreted load of something, of that something, divided by the plasma concentration of that something. So there it is. Volume of urine times the urine concentration of that thing divided by the plasma concentration of that thing. That's the clearance. And so let's look at the units. If you put the units of, mgs per mil here and mgs per mil here, which you should do, keep the units the same here, then this becomes unitless, right? This is just, the units cancel. And so the units of clearance is the same as the units of flow rate. So the units of clearance is always milliliters per minute. If you get liters per minute, it means you've done something wrong. If you get mgs per mil, you've done something wrong. The units of clearance is a flow term, milliliters per minute. Okay, what is that flow? So for some students this clicks, and for some students this becomes completely mysterious. If it becomes completely mysterious, just don't worry about it. Learn the equation and know that the units of clearance are milliliters per minute. But what that is, it's a volume flow. It's the flow of plasma that has in it the amount of X that is excreted. So if the excreted load is 0, the volume of plasma from which that X was obtained is 0. There is no clearance, no volume. Okay, so we're going to work through a couple of examples. You want to know this equation and know the units. Mils per minute? No. Milligrams per milliliter, milligrams per milliliter. If one of these is in a different unit, you've got to convert so that they are the same, because only when they are the same does this become unitless, right? You want to make this unitless so that clearance is in the same units as flow of urine, mils per minute. Remember to make these units the same. Again, I will not ask you to do arithmetic on the quiz, but I might ask you to set it up right on the quiz or the exam. Okay, so let's talk about some examples. Make this make sense. Well, what is this? This is a glomerulus, afferent arterial, efferent arterial, capillaries, tubule, right? Real simple cartoon. Filtrate, urinary fluid, urine. Bowman's capsule, Bowman's space. You all know the details. Okay, so let's take an example. The substance is not filtered and is not secreted. So if it's not filtered and it's not secreted, then none of it is going to end up in the urine, and the clearance is 0. You can convince yourself of that by plugging in the urine concentration is 0 into here, so the clearance is going to be 0. This is typical of something like protein. It's not filtered, and it's not secreted, so the clearance of albumin is typically 0. That's an easy one. Let's take us another example. Something that is freely filtered, but is completely reabsorbed, so that none of it ends up in the urine. All of the filtered material is reabsorbed, none of it ends up in the urine. So the urine concentration is 0, the clearance is 0, but this is a very different mechanism. This is an example of glucose. Glucose is small enough to be filtered, but we do not want to urinate our glucose away, right? We worked hard to get that glucose, right? That's what's keeping us alive. We do not want to excrete it, and we have evolved mechanisms for reabsorbing all of it, unless we have diabetes. We'll talk about that next week. So clearance of glucose is typically 0, but not because it's not filtered. It's freely filtered, but it's completely reabsorbed. We'll talk about that later. Okay, let's talk about a substance that is freely filtered, but is neither reabsorbed nor secreted. The only way this substance gets into the urine is via filtration. So the clearance of this stuff in mils per minute represents the rate of glomerular filtration, or the glomerular filtration rate. In other words, we can measure the glomerular filtration rate by measuring the clearance of a substance that is freely filtered and is not secreted nor reabsorbed. That's how we can measure GFR. And that's how we do measure GFR. And you're going to do this in lab this week. Or next week. Next week we've got case studies, not this week. So, for example, there is a sugar called inulin. This is not insulin. This is not insulin. This is inulin. It's a sugar that is found in some plants. And it's a sugar, so it's a small molecule. It is freely filtered, but it is not, we don't digest it either. So getting this sugar into our bloodstream requires an injection into our bloodstream. So this is hard, this is never done. I'll just be honest with you. Because it's invasive. You have to inject this sugar into the bloodstream in order to measure the glomerular filtration rate, and we don't do it. But in principle we could, because it's a sugar that is freely filtered and is not processed. And so the clearance of inulin is a good measure of the glomerular filtration rate. What we do is we measure the clearance of a byproduct of metabolism called creatinine. Muscle movement generates creatinine. We have creatinine in the blood. We have creatinine in the urine. Creatinine clearance is a pretty good estimate of the glomerular filtration rate because it is in the blood naturally. We don't have to inject it. It is freely filtered. It's small enough to be filtered, and it is hardly processed at all. So creatinine clearance is a good measure of glomerular filtration rate, and here's the normal value, 100 or so mils per minute. Okay, so this is an important concept. The clearance of creatinine or any other substance that is freely filtered and not processed tells you the glomerular filtration rate. Okay, yes?
So basically, 100% of what is filtered is excreted in the urine.
That's right. The filtered load is excreted. We haven't talked about the filtered load. Well, I'll talk about that in a minute. But yes, everything that is filtered is excreted. And in this case, well, I've used the word now. So the excreted load and the filtered load are the same thing. The filtered load, so I used it, so let me define it. So the excreted load, just to put the other one up, is the flow of urine times the urine concentration. That's the excreted load. The filtered load is the amount of filtration rate, the glomerular filtration rate, times the plasma concentration. So the filtered load of X, so those are equations to know. Remember, the excreted load of a substance is the amount volume per mils per minute times, let's just say, milligrams per mil. And the filtered load is in mils per minute. Remember, GFR is a flow term times the plasma concentration. And again, let's just call it mgs per mil. So the excreted load is in milligrams per minute, and the filtered load is in milligrams per minute. And when the excreted load is equal to the filtered load, the thing is not processed. The filtered load and the excreted load. The only way it's getting excreted is through filtering. Okay, here is a special case. So if you need to, well, let's just go through it. We have a substance that's in the plasma, in the blood. It is freely filtered, and it is actively secreted. So that all of the substance that enters the kidney leaves through the urine. None of it comes out of the kidney. So the blood flowing in has the substance. The blood flowing out of the kidney does not have the substance anymore. All of it is excreted. And so if you work through the arithmetic, you'll find that the clearance of this substance gives you a flow rate that is equal to the amount of plasma that's flowing into the kidneys. Okay? So this is a special case. I'm not going to spend too much time on it because it's just not the most important one. But if we wanted to know the flow of blood into the kidneys, we would calculate that from the clearance of a substance that is both filtered and secreted. And that substance is a natural metabolite called para-aminobutyric acid, which I will not ask you to memorize. So let's go to the next case. This is the generic case for clearance. The clearance of some substance is equal to the volume of urine flow, mils per minute, times the urine concentration of that substance, divided by the plasma concentration of that substance. This is generic now. We've gone through a couple of special cases of this equation. But now, we're just talking about sodium, potassium, calcium, urea. The clearance of these substances gives us some readout of kidney health function. So now I'm gonna take a break, but you're not, because we're gonna do some problems. You're gonna do some problems. So I want you to use the clearance equation. Here's the things to know. Here are the things to know. Creatinine, concentration, And here's the curveball that some physicians are gonna throw, and I'm not a physician, but I play one in this room. There's another unit, it's called the deciliter. So a deciliter is 100 milliliters. So you know that a liter is 1,000 milliliters. Now you know that a deciliter is 100 milliliters. So if I tell you the creatinine concentration in plasma, P sub CR, is 1 mg per deciliter, you have to convert that to mgs per mil. Okay? So when I ask you to give me an equation and to set it up, you have to pay attention to the units. Okay, so 1 mg per deciliter, 0.01 mgs per mil. Urine flow rate, 1.1. Concentration of creatinine in the urine, 0.95 mgs per mil. And this is pretty common for them to use a different unit for plasma as for urine. It's just, why do they do this? I don't know, but they do, so I do it. If it were up to me, we would never talk about a deciliter. Deciliter would have died off years ago, but it's not up to me. They still use deciliter. So, those are the lab values for this exercise. What's the clearance? What's the GFR? What's the filtered load? What's the excreted load? Go ahead and work through the arithmetic. In fact, you can go through the calculation for para-aminopurpuric acid. This is the PAH. 10 mgs per deciliter, 50 mgs per mil. Plasma concentration, urine concentration. Calculate the clearance of PAH, because we want to discuss that when I reveal the answers.